China for the introduction and have a chance to present our PhenoRob demonstrator, which is titled "Determining the Inflection Point of Plant Response to Drought and Nutrient Stresses for Efficient Crop Management." So this is a collaborative project between our departments of plant breeding and of plant nutrition, both from the INRES and IBG2 from Research Center Unit. So I would like to start acknowledging the people which are involved in this project. So today, above all, I would like to acknowledge the contribution of Nancy Salim and Sheila Krause, whose data I will mostly present today. Tobias Rox and Ian Cleon from a previous project where some of the genotypes I'll talk about are identified. Teresa Zandman and Marion Deisman from two PhD projects financed by PhenoRob, and their data are meant to flow into this demonstrator. And the three PIs, Uwe Rascher, Gabriel Schaaf, and Annalisa Mason. Okay, the two major factors conditioning the plant for the future perspective on a sustainable agriculture are world population growth, as it's shown here. The curve is growing exponentially. The blue one showing all, for the old world. And the second factor is the climate changes, which based on the predictions for the coming decades could lead to very high temperatures and low water availability for the plants, which is more emphasized in both continents, in America, Europe, around the Mediterranean, particularly Africa, and many parts of Asia and Australia as well. Well, all these leads to the necessity for production of or for development of genotypes which match with the demands or the predicted demands in the coming 30 years. And one of the topics that I'll consider today is for the production of resilient plants against stressors. And those plants must fulfill some of the criteria, and the most eminent task for plant breeders and scientists is to develop crops that have optimized shoot and root system, efficient photosynthetic activity, improved use efficiency, improved nutrient use efficiency. And as I said today, I will focus on these two factors: the water use efficiency and nitrogen use efficiency. Well, those stresses do not occur alone in the nature, are mostly combined to each other. So as we know, the green curve here shows the needs of the plant for nitrogen during the developmental stages and during the seasons from spring to the fall, when the plants grows and then until the harvesting. But as well, at the same time, it is strongly connected to the availability of the water. So depending on how much water or rain is available is in the soil. So as it's shown by the curve here, if we have a wet year, there is a more leaching and removing of the nitrogen from the vicinity of the roots. Therefore, the amount of the citrus and fertilizers is needed to be increased. How can we deal with those problems and to produce those crop isotopes? First of all, we have to understand the genetic variation in response to water and nutrient scarcities. And secondly, to analyze, or in connectivity, to analyze the plasticity of morphophysiological adaptations to water and nutrient scarcity. What is meant with that? So, well, I just very brief introduction. And what is the hypothesis when we are working on based on these projects? So it is the cut of the leaf. So as we see the cuticular, there is the upper epidermis, and there is the mesophyll cells, which actually would play an important role in the accumulation of water and nutrients. So if the water stress occurs, so the water balances will change, and this should lead to the changes in the morphological part of the cells. So the vacuoles will decrease, and the concentration of the chloroplasts will increase. All these changes lead to structural and functional changes in the cell. So the question is how to analyze those changes, how to capture them, and how to interpret them, and how to make use of those in the plant breeding, and most importantly, and as well in the plant production. Finally, to water, the stressor that I will talk about are the nutrients. We'll focus today on phosphorus, but similarly for nitrogen, sulfurs. The phosphorus, all these the scarcities of those, particularly of the phosphorus and nitrogen, lead to increase in anthocyanins. As it's shown here, there is the first reaction is the increase in the level of sugars are induced, and this leads to increase in the levels of anthocyanins. Anthocyanins are very fast visible by eye, and as it's shown on the right part of the slide, there is a strange color change if the scarcity or stress in the availability of the nutrient like nitrogen or phosphorus shown. So there is accumulation of anthocyanins and the color changes to reddish. As it's shown, as it's mentioned here, plants get blush if they are hungry. Well, all these are the prerequisites, but the importance of that is how these occur. So, and how can we analyze them? So all these are based on the specific pigments that the plant has, and each of those pigments has a specific absorption, as it's shown here. Carotenoids in the yellow one, chlorophyll green, and anthocyanins red. So each of them has a specific spectrum, but what is already shown is that this spectrum changes as soon as the plant is facing stressors. So only a few minutes after the plant is getting stressed, so it's shown by the red. So the signature of the hyperspectral signal changes, it's shown here, and that can make possible to identify those plants which are already under stress. Well, let me now come to the topic and our working hypothesis that the kinetics of water stress response in plants is not done, and the change and the switch from healthy to green unstressed plant to the weird stressed plant on the right side is not an immediate process. What we think of it is that that is a rather continuous one that occurs through several stages. So these stages are here shown. There's only a few of them, and on the left side you see the stress plant, which is very greenish. On the right side there is a plant which is wilty, but in the middle of them there are some of them which already start to have stress, but hardly visible until some time points. So what we hypothesize is that the response turning point in the temporal curve of plant response to stress, rather than the maxima, left and right, are the most relevant time points. So what can we use, or what can be the use of them, is to understand, first of all, the mechanistic base of plant response to drought or nutrient stresses, and finally they can be used for efficient plant production. So let me now show like a small cartoon. So there is a healthy leaf plant, which you can see by eye. Some after stress gets through, the plants are stressed, then they get yellow, but much more occurs in between at the molecular level, but at some point even at the morphological level, which is linked to early recognition, early signaling, and early plant response. And all this area is so far quite a black box. And this is where we want to go more by combination of invasive and non-invasive measurement. So again, the time course of the kinetics of plant response to drought stress, and if some of the non-invasive analysis can be applied, they will allow to follow in 4D the changes of the plant and able to identify the time point where the plant has the first response towards the stress. And what we think at this time point, you should start with the invasive analysis or the so-called omics to identify molecules that are firstly identified or are firstly changing on responding or recognizing the signal, and then leading to a signal transduction in the pathway. So based on this hypothesis, we have started or made the experiments. So we have identified 52 genotypes and analyzed them over 2 years under 3 replications under any semi-controlled conditions in tunnels here in the Oppelsdorf, and stress was started after flight for 12 days. So several traits were scored, and as you see on the right side, there is a graph showing the drought stress induction. And based on several morphological and physiological measurements, we could identify, so I'll just cut short the long experiment, the few genotypes which show differential response to the plant. So in green is shown one of the genotypes which has a very low wilting score. So it is classified as being resistant based on the STI values or stress tolerance index. And the red one is considered to be one of the susceptible ones having a high wilting score. So this is those genotypes that we have then selected, repeated the experiment, and tried to now follow the kinetic of the response using hyperspectral signature analysis to identify the time point when the plant starts responding to the stress. So the T extremes were selected, and they were imaged every second day after drought stress has started, from day 2 to day 13, when the changes are becoming visible, and the data analysis was performed based on this model. So on the left side, there is arbitrary spectrum, so 100% of the whole response, which is made of, here are shown only schematically, but to prepare the principle. So one portion of them, of the cells or tissues, show a very healthy leaf. So this is a normal hyperspectral images in the area where we are measuring. In B is the time points or the changes, or the leaves have already slightly changed by drought stress, and C shows the spectral of a senescent leaf. So as I mentioned at the beginning, so we are not so much interested in A and C, but rather in B. So in those the time points where the changes have started, so as much closer to A as farther to C, but as more specific for this transition phase. So based on all this analysis, based on simplest volume linearization algorithm, which actually determines 4 basic vectors for representation of data simples in complex combination. So we could identify, come to the conclusion that the first symptoms visible by hyperspectral are around day 4 to 5, whereas, as I mentioned, the symbols, the changes by visible by naked eye are first at the day 30. So it is this window here from day 4 to the day 13, which we should put in that black box of the signal transaction, of signal recognition and first transaction pathway. So let me now show a bit in another way. So the 3 genotypes, as I mentioned to you, the one of them, Damor, is the genotype which is shown to be the most tolerant one, and the Olympic the most susceptible. Interestingly enough, so shown by the long line, the divergence of this tolerant one occurs at least 2 days earlier than that of Olymp, which is indicated by the dot dashed lines. So if we have here day 4 or 5, we have in the Olymp or in the genotype, which is shown to be tolerant, in the day 6 to 7. And that we think that might allow us to not only identify an early time point, but even to differentiate genotypes which respond differently to the stress. So, as was mentioned before, I think by that, of course, the goal would be to use some devices which are easy to handle and to measure on the field, and to make fast and cheap measurements. Therefore, we will not only focusing on the images based on the hyperspectral number, we use as well the handheld device, like it is a polio pen shown on the right side of this slide, and repeat it more or less the experiment, following the response of the plants 1 up to 16 days after drought stress has started. And so again, we were able to identify that there is a time point around day 4 to 5, which is much earlier, or at least 7-8 days earlier than the the response is visible by eye. So when the wilting symptoms or the yellowish colors in the plants become by eye visible. Therefore, we think that this time window between day 4-5 to 13-14 would be very useful to understand or to analyze the mechanistic basis of the plants. So therefore, we put this window from 4 to 11 in the black box, and we would like to go farther for more analysis. So the two applications that I mentioned at the beginning are two based on this data. What we can do, we can make a guided sampling to understand the molecular mechanisms behind, and second, we can make some guided treatment. So today I focus more on this second one, which is actually based rather on pot experiments, just to understand or to show the principle. So what we do here, so we have the healthy leaves, so and we apply stress. So here from day 1. So the number or the status of the healthy leaf decreases as soon as the stress increases. So and we have 2 time points. There is one time point which we determined using the greenness index, which is at around day 5 to 6, and the second time point, which is the day 12, where you can see the symptoms by eye. So what we think, or what was the hypothesis before, is that if we start irrigation at the wilting time, at the inflection point time point, it means when the plant really starts making changes, then we'll be more able to recover the plant, or faster recover the plant, or more efficiently recover the plant. So irrigating the plant at the inflection time point will bring the plant back to the normal status faster and most efficient than by irrigating it when the damages are high. So which is at the day 12. So the plants look like it's shown here, a leaf very damaged. Therefore, the resilience or the recovery would be much more difficult if the irrigation started this way. So, and indeed, so here you have some of the experiments, Damor, one genotype, a tolerant one, Olympiad, a susceptible one. So what we have here, we have the time course of the drought stress, and we make 2 different kinds of experiments. So we water the plants at day 13, the day when we see the symptoms by eye. And as you see, there is, of course, a recovery. It is expected, but we don't reach the normal or the normal the status of the plant, which is normally treated or watered. The difference between the two is, of course, because the Damor responds differently from Olympiad. Therefore, the curve is a bit higher. So very interesting from my point of view is the C&D. When we start watering the plant at day 6, the inflection point, the point that we could identify using the sensors, the recovery, the resilience is much faster. So as you see, from day 8 or 9, we return to the normal status. So, and we think that from the production point of view, that can be very useful, because if a producer starts, so if we are able in the field to identify this time point when the plant starts getting symptoms based on the end of the sensor, and start watering before waiting that the plant shows severe symptoms, then we recover or bring the plant back to the normal physiological status much faster and with less efforts and with less cost. Okay, the second, which we have started, but we have not yet results on that, is the guided sampling and the analysis. So we'll concentrate actually, so theoretically, one can analyze all of them in transcriptomics, phonologics, or make some more phenomics at the root phenotyping. So we will mostly, if it is for molecular level, we'll concentrate, as we have already in the project described, on transcriptomics and metabolomics, and see to identify specific molecules to understand the molecular mechanism for water stress and response. Good. As I mentioned, we have done as well one nutrient stress response. Hypothesis is similar, as I said in the introduction. There are already indications in the literature that the plant gets reddish as soon as the phosphorus scarcity is detected, and we have analyzed here, or here I show 3 genotypes or synthetics, rapeseed, which shown in the lower part, and 2 diploid rapa and Oleracea are the first and the second row. So what we have done here, what Nancy has done here, is that she has treated the plants, one with normal phosphorus amounts, and the other one is having no phosphorus. So it has start suffering some phosphorus deficiency. So what we did, so of course, after some time, the symptoms are visible. You see a slowdown of the process and growth of all genotypes, reduce the leaf area, and that's negative on the overall plant height. But to make the first experiment, so as you showed that it is from the literature that the anthocyanins level increase as soon the plant is getting stressed from the phosphorus deficiency. So here I find some results on the 3 genotypes. As you can see here, the upper curve shows the kinetic over 6 days of the phosphorus deficiency, which leads to increase in anthocyanin concentrations in rapeseed leaves compared to the control, and the control is the blue one. So in all the 3 genotypes, there is a difference, and interestingly enough is that, as it was actually in the literature described, that or hypothesized that if the phosphorus deficiency leads to only a transient accumulation of anthocyanin, so it means that there might be anthocyanin after some point, as it's shown here, after the 6th week, that they can return to the normal levels. So nevertheless, we can try to use or to this data, and when the best time point is to analyze and to understand what is the difference between this what happens in the physiology of the plant at the molecular level, but as well at the same time to make a guided fertilization in the field. Okay, what actually is running at the moment is that these plants, the experiment is repeated. So again, deficiency without deficiency, but in the case now we want to follow the kinetics using the hyperspectral images analysis. And I can't show you today because the data are still on process, data processing is ongoing. Before I stop, I would like to mention as well some other ongoing and future experiments. So what I already mentioned is this identification of differential expression of specific molecules at early plant response to stressors. So the time point that we were able to identify using those sensors. So for water at type 6 in the phosphorus is maybe more complicated because the phosphorus starts from time 0. Therefore, we should see when the best time point is to analyze the differential expression of the molecules. Okay, we'll go forward with more genotypes and probably in a bigger plots, the just irrigation coping with the newly identified early trunk stress response, and to see how these under field condition gives to a better resilience performance. And yeah, that's next point is to use leaf pigment as indicators or bioindicators for demand-oriented application of fertilizers, in this case for phosphorus. Yeah, I will stop here and thank you for your attention.